Whether you're a small community seed bank focused on conserving seeds of your local area or a large international focused seed bank, it is important to manage your collections appropriately, including identifying and mitigating risks. In the last of our pre-recorded sessions, we will look at these key themes in more detail. I would also like to point out that the second talk in the session might run slightly over the break time. Hello everyone, welcome to the first lecture of this session. The first section will be delivered by Keith Manger, who will take you through what makes a seed bank, well, a seed bank. And then I will finish it off by guiding you through some things to consider when managing your collections.
So what is a seed bank? A seed bank is a container or a building that will allow you to store seeds for a set length of time. For long-term storage of wild species, we're talking hundreds or potentially thousands of years. So seed banks can vary in size. The mini seed bank, which the Millennium Seed Bank designed for schools and for the public, only cost £20, and if you follow the instructions you can bank seeds to international standards. However, if you're a larger seed bank like the Millennium Seed Bank, you're going to need a big building as featured below. So the Millennium Seed Bank was approximately £20 million, but it is the most biodiverse place on Earth and stores seeds from over 190 countries, and it is much larger. So I'd like you to consider you're designing a seed bank for your college or university to support a research project. What do you need to consider? First of all, how much seed material needs to be stored, the numbers of collections, the size of collections, and the size of the seed. So seed can vary in size enormously, from orchids at 100 microns up to palms, which could be the size of a coconut. You need to think about within each collection, which should be representative of a population, the number of seeds you're going to actually collect. So typically the MSB has between 10 and 20,000 seeds per collection, but our seed collections vary from a few, literally a few seeds, to hundreds of thousands or even millions of seeds in the case of orchids. Then you need to consider how many collections of seeds you're going to have for each species. The MSB looks for a duplication rate of 2. China, one of our partners, has opted for a germination, so for a duplication rate of 30, which is very ambitious, but will give a very statistically valued representation of the different populations of that species. You need to also consider how long the seed is to be stored. The Millennium Seed Bank is a very long-term project, so we had funding for 10 years and then that was extended to 20, but the actual vault is designed to last 500, and currently it's the largest seed bank in the world and has the capacity to store a huge proportion of the world's species. However, if you were designing a restoration project, it may only need to have a seed bank present for 10, 20 or 30 years. So for instance, open cast mining, where you collect the seeds, you store them, and then when the mining's finished, you actually grow the seeds and plant them back to restore the habitat. The type of seed you're collecting is critical. Not all seed can be stored. Orthodox seed is seed that can be dried, and if it can be dried, it can be frozen. Unfortunately, 91% of all wild species are orthodox, so the majority of seeds can be stored in seed banks. And even in wet tropical forest, we've yet to find an area with more than 50% recalcitrant species. So half of the species in a tropical forest can be orthodox and can be stored. So that talks about the seeds. When you're building a seed bank, you have to think about the available infrastructure. So is there a building you can already use? So are you going to design a seed bank in a room of a university, or are you going to build a porta cabin type seed bank out near the site where the seeds are to be needed? Is there power to the site? Do you require a generator? How many staff are going to work in the building? Can supplies be easily got to site? And also think about the security of the site. Is there a fence and maybe a security guard going to preserve what your infrastructure that you're putting in? Is it prone to earthquakes, floods or fire? All of these things need to be taken into account to build a building that has longevity and is safe to store seeds in. And perhaps the most important question is, what is your budget? So you can spend £20 or 20 million or anything in between. Once the seed's been collected and you bring it back to the seed bank, the first thing you need to do is check all your collections are properly labelled and they all link together so that the herbarium specimens actually match the seed samples. It's really important that everything is properly labelled with the accession number, that unique number, so everything can be tied in later. So you want to put the collections into the dry environment. So the top picture shows our initial dry room. The paper bags and cloth bags sit in labelled crates with the country of origin or the collection site that we've been to, and they'll sit there until the seeds are dry, and from there they'll be taken to the cleaning lab for cleaning. If you don't have a dry room, you can use other technologies. So to the bottom left is an incubator dryer. This is something that we've developed over time. It's a much cheaper option, and when it's not being used as a dryer, it can be used as a germination chamber. So it's very cost effective. A typical dry room is about £25,000, whereas an incubator dryer is about £2,500, about a tenth of the price. So the seeds are placed in the incubator dryer, and by coincidence, if you set this dryer to 18 degrees C, you will get a mean RH of about 15% relative humidity in the chamber, and the air is recirculated over a chilled coil. So this is actually condensing the moisture and taking it out of the chamber, and it works extremely well. And it can dry quite a lot of seed and is very suitable for a small seed bank. The Malian and Malawi seed banks shown earlier in the presentation have incubator dryers. The most basic type of dryer is a drum dryer. So this is a metal or plastic drum, usually about 60 litres, with about 20% of silica gel at the bottom with indicator in it. You need to monitor the silica gel to make sure it's dry at about 10 to 15% relative humidity. But if you do that, you can place bags in that drum and seal it, and the seeds will dry down. You do need to monitor them though. This is the most basic form of drying, but if done properly, can produce seeds to international standard. Another piece of critical equipment is the relative humidity hygrometer. This in the picture is shown a rotronic hygrometer. This is a non-destructive way of measuring seed moisture. So after seed has been dried and equilibrated to 15% relative humidity, a random sample can be placed in the hygrometer, and within a matter of minutes, and certainly no more than 20 minutes, you'll get a stable reading. And that will show you the relative humidity of the air above the seed, and that represents the moisture status of the seed for most seeds. Very few seeds do not perform well with hygrometry. We're looking for 15% relative humidity, and if the seeds after a few minutes equilibrate to 15, we know they're fully dried. Once the seeds are dry and are ready for banking, we need to estimate their quantity. We do that using a digital balance of a suitable size. So we generally use 4 to 5 place balances which are calibrated. We will weigh the total seed collection, and then we will count 5 lots of 50 seed from the collection and weigh those and work out an average. Divide the average into the total, and you'll then get your total number of seeds, and that's recorded on our database. The seeds are then placed in appropriate containers for banking at -20. On the top left, you'll see our range of glass bottles, clamp-topped 3 litre, 1 litre and 500 ml storage jars, blue-topped 100 ml shot bottles, 30 ml universals and 2 ml vials, which are also stored in universals for ease. Each of those has a special label, which will not come off at -20 degrees C. This has a unique accession code for the collection, a location code so we know where it's to be stored, and that's also enshrined in a barcode which can allow for automation. Below that is our foil bag sealer and foil bags to the left. So foil bags are used in our base collections, glass bottles in our active. The foil bags we purchase in a range of sizes, and they're specific for seeds. And the sizes are from A3 to A7, A7 being the smallest, and each size halves. We choose the appropriate size container, place the seed in it, exclude most of the air, and then place it in the heat sealer and obtain a 1 centimetre thick crimped seal, which is very, very airtight. The advantage of foil packets is that if you drop them, they do not break. They are very light, and most seed banks use trilaminate foil, which is what we're using. So it's the most commonly used seed bank container. On the right hand side you will see some 1 gram silica gel sachets in perforated bags. We place one of these in each container. The silica gel beads have an indicator called methyl violet, which is harmless to seeds, and when it's wet it's green, when it's dry it's yellow. Unfortunately for us it changes that colour between 15 and 20% relative humidity, so when that bag goes in with the seeds it equilibrates to the same relative humidity of the seeds, and at a glance we can tell whether the seeds are too wet or not. All the silica gel sachets should be yellow. The equipment shown on this slide are incubators. We use cyclic incubators for doing dual temperatures, mimicking the temperatures you might get during daytime and nighttime. The temperatures are controlled to within 1 degree of the whole incubator, and the incubators are fan assisted. They're very low energy incubators with LED lights, and they're called white LED lights, and we generally set them at 5 degrees C increments. So you can set them at a constant temperature of say 20 degrees C with the lights on and off for different photo periods. Typically we use 12-12 photo periods for plants that grow between the 2 tropics and around the equatorial regions, and 8-16 photo periods for the temperate regions. And you can also have alternating temperatures, so we can switch to, we've got 2 controllers on them, and we can switch between the controllers with a time clock, so you could have 8-16 with the lights on for 8 hours and change the temperatures at night and day accordingly. They're very useful incubators in different sizes. The white ones are typically 250 to 280 litres capacity. The big silver ones in the back are 600 litres capacity. For growing seeds in a laboratory, we don't use soil, we use typically agar. Agar is a seaweed extract, it's sterile, and we use 1% water agar. And to this we can add growth compounds such as gibberellic acid, which is a hormone, or nitrate for promoting germination, and even smoke compounds for fooling seeds to think there's been a fire. And it's a very useful media for doing that. The agar forms a jelly and the seeds sit on top. They absorb water in vapor phase very gently, so they don't sit in liquid water, and generally you get very good seed germination. You can't generally grow plants in agar. They would be transferred to the glasshouse for if you need on growing. Our partners have also helped us move towards sand on occasions as well, so it's sterilized, clean, washed sand. And sand with a little bit of water can be a very good growth media, and it allows for good root development and is particularly good for larger seeds. We rarely use germination paper because paper very often dries out quite quickly, and we do vast numbers of germination tests, and so it takes us a long time to keep perfect conditions. It is cited as a germination media and sometimes used for research, but we mostly use agar and on occasions sand. When we're scoring germination tests, there's the possibility of also growing molds, because although seeds have been dried and frozen, mold spores or fungal spores also survive drying and freezing. So seeds aren't sterile. And when you put seeds on agar, some sugars can leach out from the endosperm of the seeds, providing a perfect growth medium. For this reason, we score germination tests in a laminar flow hood. So the one pictured, the operator sits in front of it, the air is sucked across the face and up into the filter at the top, making sure that any fungal spores that are released are trapped in a HEPA filter, protecting the person underneath. And typically we will sterilize the surface of where we're working throughout the labs with a disinfectant before and after use to reduce infection. Seeds can be stored at a range of temperatures. A fridge at 4 degrees C can be used as an active short-term store. If you are going to store seed at this temperature, ensure the seed is in absolutely hermetically sealing containers, because any fridge at 4 degrees C is at 100% relative humidity, and it's very important to keep orthodox seed dry. We don't use 4 degree storage at the Millennium Seed Bank, nor do we use permafrost because we don't need to. Permafrost is better than 4 degrees, but is rarely used in truth. But it is obviously a natural area, so there's no electricity and could be used as a low energy solution. Generally, freezers are used at -20. These can be upright freezers in labs or -20 cauldrons for large seed banks like the Millennium Seed Bank. If you're using the latter, you need cold room clothing. You need to be careful because -20 is a hazardous temperature for humans. We can only stay in there even with cold room clothing for 15 minutes. And last is liquid nitrogen. So we use liquid nitrogen for cryopreservation of recalcitrant seed in our research facility. That's going to be discussed elsewhere on the course. But we also use it to duplicate orthodox seeds that are short-lived. There is an advantage because the flash freezing down to -190 degrees C allows short-lived species to be kept for longer. It is important if you're using liquid nitrogen to have a liquid nitrogen alarm that's pictured above, because liquid nitrogen contained in a room is potentially hazardous. The room needs to be ventilated constantly, and if there's a mass release of liquid nitrogen, the oxygen level will drop very quickly and you'll need an emergency fan to cut on and an alarm to go off to evacuate the area. So liquid nitrogen is a very good resource for long-term storage of short-lived species or for recalcitrants, but you need to use it with care.
Thank you, Keith. So alongside the range of drying, cleaning, germination and storage equipment that Keith has gone through, equally as important is a robust system for monitoring the size and health of your collection. A good data management system will ensure effective decisions can be made for your collection in the long term. In the last lecture of this session, we will show you an example of a data management system using Brahms. For now, I will go through key things to consider when managing your seed bank and your collection. Before you go ahead and build your seed bank and start collecting, it is important that you have a clear idea of how the collection is going to be managed. During every stage of the seed banking process, there will be a number of management decisions that have to be made. What do we do, for example, when all seeds are empty or infested? Or if they come back to the bank and mature? Or what happens if some collections fail germination testing or viability testing? Or simply if we have a mixed collection? These should all be discussed thoroughly with your teams and a clear standard operating procedure in place for implementation. It is important that any policy or SOP needs to be reviewed regularly and adjusted accordingly to the needs of your collection. Here is an example of the MSB's decision tree for collections. We look at about 400 to 500 seeds as a tipping point, as this would mean about 200 will be able to be in our long-term storage or our base collection and should not be worked on. And then we'll have about 100 for distribution. As we are a place where many partners duplicate their collections, we also look to see if the original collection in country is sufficient in number. This is usually 10,000 seeds. If there is a sufficient collection in place, then we would consider that conservation collection is in place for that species. If there is none, then we would usually prioritize recollection if possible. Your seed bank decision tree can look very similar or very different to this, depending on the type and purpose of the collection you hold. The important thing is to have a clear one in place and reassess to see if decisions that are made are appropriate. Another consideration when monitoring your collection is distribution. For many of the collections we hold, our partners consent to these being made available for use by third parties, usually for non-commercial purposes like research. Sometimes these go with or without prior permission, although we tend to always ask anyway. Some of you might be kind of long-term MSB partners, so you may have come across an email from either myself or Eleanor asking whether the collection you collected, sometimes maybe 10 or 15 years ago, could be used for research. We currently offer this through an online portal called the Seed List. However, prior informed consent is not the only thing we consider. A full distribution policy should be in place and a list of criteria on it should be met before a collection is suitable for distribution. At the MSB, a collection must be big enough, alive, fully identified and under agreement with the donor organization before it can even make it onto the seed list. Finally, we also need to think about risk and managing the risk to our collections. A risk is a hazard that we have identified which will negatively impact something. Alongside the risk, the likelihood and severity of that risk also needs to be considered. There are many different types of risks, but most come under three broad headings. Natural risk, like the risk of collections catching on fire or the seed bank being flooded. Financial risk, for example, if your seed bank is purely project funded, there is a risk that routine procedures like germination can cease after project funding ends. And then political risk, which can be risk from uprising, or it could be unwanted exploitation of collections for commercial purposes. The magnitude of each risk can vary depending on your individual circumstances. For example, a nationally run seed bank in Geneva will have a very different risk management strategy compared to an NGO-run seed bank in Afghanistan, and so on. But all risk can be managed through identifying appropriate mitigation action. In this example, we have identified a big risk of fire to our collections, so we can put in place early warning systems and fire doors and ensure that all personnel are trained in fire safety and evacuation processes. We also identified that there will be limited funding, so we can mitigate this by duplicating part of our collection to another seed bank where routine procedures can take place more securely. Finally, we know that we have commercially valuable collections, so we need to ensure that strong material transfer agreements and access and benefit sharing legislation is in place. Thank you for listening. If you have any questions, please do join us for the discussion session after the break, or get in touch through our emails. The next lecture will be by Naomi Carvey on the MSB Data Warehouse.
Hello, and I wish you a very warm welcome to today's training course on the MSBP Data Warehouse. My name's Naomi Carvey. I've been working for Q for around 6 years now. I'm a data and database specialist. In today's session, we will be looking at the website of the Millennium Seed Bank Partnership and the MSBP Data Warehouse database. As you know, today's session has been pre-recorded, but I will be watching the presentation alongside you during the training course, and I'll be available for any questions you may have during the session or afterwards. And equally, if you have any questions after the training course or just want to say hello, please do get in touch. My email address is on screen now. Okay, great, let's get started. Here we have the homepage of the Millennium Seed Bank Partnership website. The web pages are organised under various headings which you can see here. And from this homepage there is also a button to jump directly to explore the database here. As with all of the screens that I'm going to show you in this presentation, you can find the appropriate web address up here under the slide title. Now I've gone to the training menu and I've selected the resources page, and here you can find a variety of different resources, 3 of which I'd like to draw your attention to in this video. Firstly, technical information sheets, which you can download for your own reference. Secondly, technical videos. And thirdly, and off screen here, you can find a link to the MSBP Seed Conservation Standards document. When we talk about the MSBP Data Warehouse, we are primarily referring to the massive database of seed collections and associated data. You can find that under the seed data heading, which I'm pointing out here. And under this heading, you can also find some other useful information, for example, base lists and technical information sheets, some of which we'll have a look at in subsequent slides. To access species base lists, you can go to Seed Data Base Lists, and there are 2 different versions of the base lists available. Firstly, you can download a base list for the whole MSBP network, so not just the Millennium Seed Bank, but for seeds located worldwide throughout the partnership. And secondly, down here, you can download base lists of species and collections just for seeds stored at the Millennium Seed Bank. Also on this page, there are some instructions about how you can check a species target list against species collected through the MSBP Global Partnership, and I update those species-based lists every 3 months or so. The MSBP Data Warehouse is not currently open access, and that's to do with some of the legal conditions associated with some of the seed collections within the database. Hopefully one day we will make the data warehouse fully open access, but we've got some work to do before we can get to that point. Access is available to anybody who works for an organisation within the Millennium Seed Bank Partnership. And to get access, if you go to the registration page, and as usual, the link is here under the title. If you register, if you fill in the mandatory fields through registration and then email me on this address here, then I'll know to give you access, and upon registration I'll confirm your access and send you the data use conditions. Once you've logged on to the data warehouse, you'll see a screen that looks a little bit like this. The default table which the database opens in is the seed accessions table. You can see that here. There are 4 tables: seed accessions, germination tests, taxa and botanical records, and there are approximately 193,000 seed collections in the online database. In this table, seed accessions, you'll find all the field data, all the storage data, for example, seed bank and location, etc. You'll find the X-ray test data, as well as a few other bits and pieces. If you click on the yellow drop-down arrow here, you can change to one of those other 4 tables, and here we see germination tests. In this table, you can see very detailed information about conditions, about test steps, and about test results. I'm going to show you one more of these 4 tables. Here is the taxa table. In this table, you'll find approximately 57,000 taxon records, which includes subspecies and varieties, etc. And this is also the table where you can find the IUCN global categories. Now I'm going to talk a bit about navigating the data warehouse and finding the data that you're interested in. So you can see on the left here we have the tree views, and you can use those to filter on the data you're interested in. There are 3 tree views: collectors, geography, and taxa. Here, collectors, geography, and taxa. And you can see I've expanded the results for geography, and I've typed in the letter I in the filter box here, and the results have auto-updated with just countries beginning with the letter I. Within that, I have ticked Indonesia, and note here you can see that there is one active filter. That's the filter on Indonesia, and I'm going to return to talk about managing filters in a subsequent slide. After Indonesia, you can see the letter 8. Now that means that there are 8 regions under the country Indonesia. So if I were to click on that little plus sign there, we would see 8 regions under Indonesia, for example, Bali. And you can see that I've done that in the taxa tree at the bottom left of the screen here. So for the family Actinidiaceae, there are 2 genera, which we can see here. I've expanded that. And if I were to click on, for example, Actinidia, there would be 6 species under that genus. In this screen, I've clicked on Manage Filters, when you open the data warehouse, you will by default see the tree filters, but here I've switched to click on Manage Filters. And you can see that on this screen there are 3 active filters on the countries Indonesia, Thailand and Vietnam. Now I have 2 options if I want to turn those off. I can simply click in these little boxes here to turn them off and on, to toggle between off and on, but I'm not deleting the filter by that option. Or alternatively, I could click on these red crosses on the right hand side and that will delete the filter completely. If you're working in the data warehouse and the results aren't what you expect, one of the first things I would recommend is to check your filters, because it could be that you've accidentally got too many filters or things haven't updated in quite the way that you wanted. So you can often get the results you want by managing your filters. Now we're going to have a look at some of the icons on the toolbar which you can help to navigate the data warehouse and find the data you're interested in. So if we start from the left hand side here, you've got this button to select visible columns, which will open a little side panel on the right hand side of the screen. And in the next slide we can see what that looks like in action. The next option along the little magnifying glass icon is to view all data for the current record only, and again that will open in a panel on the right hand side of the screen. The next icon is to summarize data. So for example, if you wanted to see all families within the data that you've filtered on, typically this would be used for filtered data. You can see how many for each family you have. The button here is for a simple filter, and then the little Excel sign here means you can download all the data that you're currently filtered on to Excel. And on the right hand side of the screen here you have various buttons to open that taxa in a different website, a different web page. So for example, here this case sign would open that taxa within the Plants of the World online portal. And again, we'll see what that looks like in subsequent slides. So in this screen, we are in the seed accessions table. And I have clicked on the Select Visible Columns button, and a side panel has opened here on the right hand side of the screen, which is showing me all of the columns that are within the seed accessions table, because not all of those columns are shown by default when you open the database. There are very, very many columns, and a few of them we don't use in the data warehouse, so they might always be blank, for example. So I have pre-filtered, if you like, on the columns that I think are most useful for most people. But please do be aware that there are more fields within the data warehouse, so you might like to have a look and see if there's something else which is of particular interest to you. And that goes for all of the 4 tables for seed accessions, botanical records, taxa, and germination tests. I nearly forgot the 4th one there. So if we see here, for example, the first column that we can see in this table is seed bank. And we can see that that's ticked there. So the tick means that it's visible. There is in fact another column before that called DB, standing for database. And I haven't selected that one because the database is the data warehouse, so I didn't feel that it's particularly useful within this database. But do be aware that there are more columns that you can filter on there. And be aware that because there are so many columns, you can't see everything in one view. It would be tiny, tiny, tiny. So do remember to scroll to the right down here so that you can see all of the data columns that are selected. And one last thing to say about this, the columns that you select or that have been pre-selected for you is what is downloaded to Excel if you use the Download to Excel button. In this slide, we are having a look at the View All Data for Current Record button. I've selected one record, in this case a seed collection of Dalbergia cultrata, and I've clicked on this View All Data for Current Record button, and you can see a side panel has opened in the right hand side of the screen, and it's giving me all of the information for that one seed collection, which for most people is easier to see that in this one panel on the right hand side rather than to scroll across and try and see all of that data within the table. So in this screen, we're going to have a look at the Summarize Data button. So I have filtered on the seed collections that are of interest to me. And say, for example, I want to know which families are within my data set and how many collections of each family I have. I can click on any family within the list, not the title, but any of the seed collections records results. So here you can see I've clicked on Asteraceae, and then I clicked on this Summarize Data button. And again, within this right-hand panel, it's telling me how many of each family I have. So you can see my top result is Mosaicae. There are 78 collections of Mosaicae within the data that I've filtered on. If I wish, I can download that summary to Excel using this Excel button within the panel, not the one within the data set here, but this one here on the right. And I can also use those results to filter further. So for example, if I were only interested in the top 3 families, I could tick the boxes by Mosaicae, Poaceae and Fabaceae in this example, and then click this filter button here, and it would filter down onto just those 3 families. In this slide, we can contrast that with how the simple filter button works. So in this example, I've clicked on Asteraceae as per the previous slide, but if I click on this filter button here, it will return just the Asteraceae records within the data that I've filtered on. So in this screen, we're going to download data to Excel. So I have applied some filters and I'm looking here at 72 seed collections, and I would like to download that data to Excel. And I can do that easily by clicking on this little Excel button here. If you download data to Excel, you can use that freely within your organization, but please do not share it publicly. If you'd like to use the data in any published works, for example papers, please cite the data warehouse, and you can find out how to do that within our terms of use on the website. I'd like now to draw your attention to quite an important field within the data warehouse, and that is the TTH field here, the second column within this view on screen. TTH stands for Transferred to History, and that basically is a flag of a low quality collection. There's a few reasons why we might transfer a collection to history, mainly if it's failed germination tests, or if the seeds have been proved to be mainly empty or infested in an X-ray test. if those seeds are immature. Perhaps all the seeds are dead, and they were particularly concerned about post-harvest handling. So, for example, perhaps the seeds have been collected and they were perfectly fine, but then they were left in a hot car and unfortunately have died. If all the seeds have been used up, or there were very, very few seeds in that collection, too few for it to be useful for seed banking purposes. And just occasionally we also get that no seeds have been collected and other reproductive structures have been collected by accident instead. So when you are analysing your data, please be aware of this column because for some purposes you may need to exclude collections that have been transferred to history. Now let's have a look at mapping in the data warehouse. So here I have my 72 collections from Indonesia, for example. I've already filtered on those within the seed accessions table, and I've clicked on the Maps option here, which opens this Google Maps interface that you can see in the slide here. There are quite a few different options within the mapping module of the data warehouse. You can do things like see summaries for each data, which you can see in the right-hand side panel here. And you can click on, for example, this zoom to location button. You can change the base maps. For example, I don't know if you wanted street view or you wanted black and white, for example, as well as all the usual zooming in and out, etc. Here, let's look at a different screen within the mapping module. So here I've clicked on data display rather than data summaries, which was the last slide that we looked at. And one thing I'd like to draw your attention to in this slide first is I have unselected use map marker clustering, which is the default, and that groups collections together to give you more of a sort of heat effect. Personally, I don't like it, so I will always deselect it whenever I go into the map screen. And that enables you to see all of the individual data points, which you can see here, for example. Now, one quite neat bit of functionality within the data warehouse is you can filter by IUCN categories. So here in the taxa table, I've selected only collections with an IUCN global rating. I've filtered on them, and here I've zoomed into Southern Africa and Madagascar, and you can see that the collections here have different colours based on their IUCN category ratings. For example, yellow is vulnerable, orange is endangered, and red is critically endangered. So for our final screen on the maps function within the data warehouse, I would like to show you that you can create heat maps within the mapping options. So here we can see I've applied a heat map to Australia, which is showing us that the greatest species diversity is in southeastern Australia down here and also down in Tasmania. And one other thing that's quite interesting that you can see within this screenshot is fuzzy mapping, what we call fuzzy mapping. So can you see over in this left-hand corner in Western Australia that there's like a grid effect? That's because I have applied what we call fuzzy mapping. Now, some partners, particularly those that work primarily with threatened species, as is the case here in Western Australia, Some partners do not want the exact location of those collections to be disclosed online, so they opt for something called fuzzy mapping. So the benefit of this is that you can still see that seed collection on a map, but that is not the exact location. It gives you a rough idea, but you could not recollect, for example, from the location that you can see here. One thing to bear in mind about this is where you have fuzzy mapping, there could be many collections on top of each other, and you can't tell that from the map. So you might see one dot, but there might actually be several, even possibly 100 collections under that data point. Finally, let's look at X-ray images within the data warehouse. So in this screen, I have filtered on the species Daucus carota, and you can see that I have clicked on the images button here, and some X-ray images are available. These are very high resolution images, and you can zoom in to really quite a fine level of detail within those. There are just over 2,000 images currently available on the Data Warehouse website. We do have many more than that for collections stored at the Millennium Seed Bank, and we hope sometime in the future to release many, many more of these X-ray images onto the database.
Many thanks, Naomi. I hope you found our sessions useful. If you have any questions and are unable to come to the next session, please do get in touch. For general seed banking queries, you can email either myself or Eleanor. For any specific queries regarding the data warehouse, Naomi is always on hand to help. We will now have a 15-minute break, after which we will start the live session. This will consist of a general Q&A and themed breakout rooms. Thank you.